I was a biochemist. Yes. Well, thank you, Diego. All right, so this is my second lecture. Yesterday I gave one on kinetics. Today I'm going to be talking a little bit about how to use NMR, nuclear magnetic resonance, to do some experiments that are important for protein folding. Before doing that, I'm going to give you a little bit of an introduction on the rationale for doing these experiments first, and then a little bit on the technique. It may become a little bit too basic, so you think that what I'm saying you already know, let me know, and I will move more quickly, okay? And I'm sure that I'm not going to be able to cover everything that I have here, but I just we'll skip through some of the areas as we go. So anyway, so so here the main idea is to try to get information about protein folding mechanisms and the interactions that play a role in that using a structural technique like nuclear magnetic resonance, and also be able to use that kind of information to compare with simulations. And that's probably the part most relevant for the majority of people in this audience. So I wish to be able to get to there and talk more about this. So, as Diego said, originally when somebody looks at this problem from a biochemical point of view, which is what people were doing in the 70s, 80s, and even 90s, the protein folding problem was described as something like this, okay, which is a very simple process with two rate constants. I was talking about this yesterday. You look at this from a more physical point of view, it looks like something like this, in which you have something that may behave like a reaction coordinate, a projection of the landscape into this, and then you have free energy, and then you have something with a barrier separated into states, okay? So if you look at this from the point of view of the populations, the system behaves like a first order transition, which is what you have here. This is a different order parameter, but still, you can assume it's the same. Here you are changing temperature, plotting probability, and what you see is that when you are in native conditions, you have one population just here. When you are in unfolding conditions, you have another one, and when you are at the folding temperature, you have 50-50, and there is nothing in between, okay? So this is something that, as I say, that biochemists find very attractive because it's simple, and then you can get your numbers from the experiments. But what this tells you really is that the systems, if proteins work like this, there is not much point in trying to do a very high resolution NMR analysis, because the idea is that no matter what property of the protein you look at, you will see something very similar. So it's like an overkill, right? So you do lots of experiments, very painful experiments, and you gain nothing. And I'm saying this because this is probably why people were not doing the experiments that I'm going to be telling you about later. Just because they thought, wait, we don't want to spend 2 or 3 years of my life doing something to find repeatedly the same process, right? Doesn't make much sense. Now, when I started working on these ideas, I was interested in this downhill folding predictions from the energy landscape, Peter, Jose, and all these people. And at the time, everybody was focusing on the kinetics and saying, okay, the kinetics had to be non-exponential, stretch exponentials, and things like that. But then, at the same time, I realized that, this kind of behavior, in which you don't have a free energy barrier separating the two states, if it goes to the stream, it becomes a second order transition like this. And then the implications are also in the thermodynamics, right? So you have a system in which basically you are going from native-like in stable conditions to unfolded-like. You are not as native and you're not as unfolded because there is some degree of coupling, but not as much. And then what you have is this kind of continuous light transition. And when you have this kind of behavior, there are very important implications for equilibrium because what it means, depending on the property that you're looking at, you will find different thermodynamics. And everybody doing simulations here is probably familiar with this. But from an experimentalist point of view, people didn't realize that's the case. And actually, at the time, I thought this was a stream. I mean, this was going to only show up in experiments when you have the stream case in which there is no barrier at any condition, which is what we call global downhill. Okay, so we looked at this as a proof of principle, and this small protein, this is really old stuff. It's a very small protein. It has a very non-cooperative behavior. It has all these different, we look at this with different probes. We did a statistical mechanical modeling of the process, and then we found that, the system behaved, and this is basically the modeling that comes out from feeding the data, the experimental data. It looked like it was downhill, and it looked like you had this kind of gradual process. And of course, this was interesting because then, with a system like this, it makes sense to try to look at this with a high-resolution technique like NMR, right? So, so we did that, and this is also old stuff. I'm not going to talk much about it. I just using it as a means of introduction. So when we were doing NMR in this little protein, which wasn't particularly easy because it's a protein that we produce chemically, which means we couldn't have isotopically labeled samples and things like that. We could find very complicated behavior, each different atom doing something different. And of course, this gave rise to this idea of being able to look at the process with a high resolution technique and be able to map what every different atom in the protein does from experiments. Okay? And these are very basic, simple experiments, titration experiments in which you just look at the protein by NMR and change the conditions and do the experiments again. And it's like a simple titration experiment that you do in analytical chemistry. Okay? Now, all of that was interesting, but as I say, at the point, we thought this was specific of this global downhill scenario. And the question, how useful this kind of approach could be for other type of proteins. Or in another way, how common is this downhill folding business and how close to that other proteins are? Because, the big idea was, okay, maybe that protein behaves like that, but it's like a very special, unique protein, and who cares, right? Because you can use this technique for anything else. So, so then we started looking at this problem from that point of view, and this is what, in order to do this, we used some very simple statistical mechanical models. These are mean field models, and they are very, very simple. I'm just going to go quickly through that. I didn't have time yesterday. If anybody has questions, let me know. Just the only thing I want to say here is these are really, really simple models that we are trying to use to interpret experimental data directly. So we are not trying to understand the mechanism or the process at the molecular level. We're just trying to see whether we can get something beyond this two-state model that people normally use. Okay. So the model that we have used for this is something as simple as that. It's really, really, really simple. So you have an order parameter, which we also think is a reaction coordinate, which we call nativeness, that is basically a local order parameter. So it tells you how native-like your dihedral angles on the protein are. So this goes for each peptide bond. So everything scales to the number of peptide bonds that you have in your protein, and then you have all this function, which is it represents the entropy of mixing plus the difference in entropy from being folded versus being unfolded. Okay, and then so that's for the entropy, and all of this is defined. So this is coming just from a simple idea, which assumes that this values of nativeness is a microcanonical ensemble, while the nativeness is a macrocanonical ensemble. So really, really simple stuff. Anyway, so for the enthalpy, you describe this as a Markov process. So that means that you assume that the rate of breaking interactions is constant with the order parameter. And then you can define two types of interactions, the ones that are local, that are defined by one particular curvature of this function. By the way, a Markov process is exponential. And then you have a non-local term, which tells you about tertiary interactions in a way. So those are the terms. You can get the free energy then like this, very, very simple. And the way we did this analysis is assuming this delta S per residue is fixed to this value, and this is coming from experiments in calorimetry, so that seems to be a pretty well-determined number, and it's roughly constant, okay? We assume it's always the same. You just have two parameters, which are this and this, because these curvatures, we fix them, okay? So the way this model looks like And by the way, and then you represent the kinetics as diffusion on this the free energy surface that comes from here, right? So yes, with a constant refactor. Yeah, the idea is to have something that is procedurally as simple as a two-state model, but it gives you much more because you have a free energy surface, and therefore you have a barrier that changes height and position as well. So you can use this directly to fit experiments. Because remember, when you're fitting experiments, you have to use the model that has the fewest number of parameters. And of course, if you go to the string case, you have a model that doesn't tell you anything about what's going on. So it's not that useful. Anyway, so this model gives you this behavior. I think I showed this picture yesterday. So this is the order parameter, is defined as a probability in a way. So it goes from 0 to 1. And this is the average for all the peptide bonds in the protein. Free energy. The entropy function has this shape. And of course, the maximum in entropy is over here because there is a much higher cost of fixing the dihedral angles in native than in being unfolded. But obviously there is some entropy of mixing, so that means there's not over there. Then the stabilization enthalpy has this exponential decay because it's a Markov process, and because they have different curvature, when you put this into the free energy, it gives you a barrier. Okay, you have a broad well for the unfolded state, you have a narrow well for the folded state. You have a barrier that is coming from this entropy enthalpy decompensation, which is exactly what the energy landscape was saying for a long time. Now, this function here, the blue, basically is coming from the sum of two different functions. One is for the non-local, which is steeper, and one that is the local one, which is broader. And this just has to do with the fact that when you have a local interactions, you don't have to fix so many degrees of freedom. When you have no local interactions, you fix many. So that means that the rate of breaking these interactions is much faster than this one when you're coming from the native state. Okay, so that's the idea. So what we did with this is now we took this model and we picked 65 single domain proteins that people have studied experimentally. We took their experiments and we fit the model. Rather than using a fit to system model, we used this model. And then we could reproduce this well. And what we found was kind of interesting. First of all, we found that there is a very strong correlation between this delta H no local and this delta H local. It's an anti-correlation. And if you go, this line, which gives you a correlation line of 0.97, is what you get when the total delta H per residue is constant. So what this is basically per residue, okay? So what this is basically telling you, this line, is that the delta H per residue is almost constant for all these proteins. So it's almost no change, right? And it amounts to basically a free energy difference of about 0.35 kilojoules per mole per residue in free energy. So that means there is a very strong scaling of both. And so the fluctuations that you see here are the differences in structure and sequence, very little, okay? But the important thing from what I was trying to say is that in this kind of analysis, what you find is there is a group of proteins here that are high content of local interactions, low non-local. This is kind of the downhill folding regime, right? All these proteins, according to the model, when you look at the free energy surfaces you get from fitting the data, they all look downhill. Okay, so that was the first point, which is interesting. So that means that it's not only one protein, which was this one here. There is a whole other set of proteins that people looked at that they are also downhill. That means that this kind of analysis could be used for them as well. But we were also interested in seeing, okay, what happens over here, right? Because these are clearly downhill, but when you are over there, maybe you still have some of these features that you can see experimentally. Okay, so you use the same type of model to look into that. And this is basically an analysis with the same model in which the only thing we do, this is completely theoretical, okay? The only thing we do here is change the barrier height. You can see models in which you have a high barrier of roughly 15 kT, barriers that are almost disappearing, okay? And this is the way the equilibrium probability function looks like. And this is what the kinetics, the chevron plots that we were talking about yesterday look like. So what you see, obviously, is when the barrier is higher, the rates are slower, no surprise. It still looks like a chevron plot, but the interesting observation is that as we go into smaller barriers and faster kinetics, also the chevron plot is getting flatter, okay? And this is interesting because from an experimental point of view, you always have to compare relative parameters, right? Because anything scales with size, sequence, everything else. So you want to try to find something interesting about the behavior of the data. You have to find always relative terms that you can compare. So here in relative terms, all these systems are the same, with the only difference being the barrier. And when you increase the barrier, you see faster, but obviously we don't know how fast, so that doesn't tell you much. But you also see that it gets flatter. And the flatness is something that you can analyze empirically from data that people get when they do their standard experiments. So one parameter that you could look at then is this M value. I was talking about this yesterday. You remember this is the slopes of the chevron plot, the sum of the two. In equilibrium, what you see here is that as you change the barrier, this is the barrier, the maximal barrier at the folding temperature, at the midpoint. Okay, so the barrier is high. You know, the M value, this is in relative terms because we have theoretical analysis, right? So we know it's maximum, so there is nothing missing there. But as we go into smaller barriers, it's getting flatter, which is what I was telling you about. Okay, this is something that is not particularly useful for an experimental point of view, because you could always argue, okay, this protein is flatter than this other one because of the properties. But what is interesting is that when you look now at, this is what you get from the equilibrium experiment. When you look at this kinetically, you see the same effect, but it's more marked. Okay, so now you actually have a parameter that's interesting empirically, because now you can compare the ratio, the M kin versus M ec, which is here, and what you can see that it follows a very specific trend, right? So it's very close to 1 when you have a high barrier, because that means the system is two-state-like, fine. But as you go to lower barriers, and this, remember, is the maximum barrier, is at the midpoint, then this number decreases. And this is a number that you can compare in any experiment, because you can do the kinetic experiment, you can do the Coherence experiment, is relative, and then you can see whether things change or not. Okay, so when you keep doing the analysis now and you look at the free energy surfaces you get from here, now what I'm plotting here is this midpoint barrier, is the same in this graph, but here I'm putting the barrier that you will find in native flight conditions. Okay, and this has to do with the type 0 versus type 1 scenarios and everything else. So when you do this plot here, what you see is that there is a range of behaviors in which both of them are downhill, right? So there's obviously downhill when the bar in physiological native flight conditions. There is also no barrier at the midpoint, and this is what we call the global downhill, which is where BBL was. Okay. Now, if we go over here, we still don't have a barrier in native flight conditions, but then there is a barrier actually at the midpoint. So this is a regime in which the protein is downhill in native conditions, but has a little barrier at the folding temperature. And then you go into this regime in which there is significant barriers on both. So that's the two-state-like regime. And then there is also this thing which we call the twilight zone, because there is a barrier, significant barrier at the midpoint, but there is a very small barrier in native flight conditions. Okay. And now the idea here is to try to look at, with this kind of scheme, to look at all these proteins that people have studied and see how they classify. And again, the idea here, the rationale is to see whether the approach that I'm going to be telling you about is something that can be used for many proteins, not just string cases. So what I have here is the plot of this M kin versus M ec, and this is the rate that you find at the midpoint, okay, from the chevron plots. So this has to do with the barrier to some degree, height, but of course this is from experiments. The black line is what we get from the theoretical analysis, and all the dots are basically coming from experiments. And what I'm plotting here with different colors are different mutants made on different types of proteins. So each color is one type of protein. All the points represent different mutations, and then you do the chevron plot, and then you look at the kinetics, the slopes from the kinetics, the slope from the equilibrium, and then you compare. And this line here is not a fit. This just comes from the analysis directly, right? So when you're plotting these numbers, what you see, obviously there is quite a lot of noise, especially over here. Why? Because sometimes you make a mutation and the slopes change for other reasons. You change the structure of the unfolded state, you make the protein more open, things like that. But in general, you can see a very clear in the rate, but also in the M kin normalized. Okay, right. So what's the implication for this plot? Well, the implication is that some of these proteins are these super fast folders that I was telling you about, but many of these proteins are proteins that people have classified as two-state-like. And according to this analysis, they should be more like downhill in water or twilight zone-like. So proteins that will have some kind of complex behavior in equilibrium, in principle. So, of course, the implication for this is that we should be able to use this type of analysis, beyond downhill folders, which would be right here. Okay. So we did the same thing looking at calorimetry, and here we're talking about the specific heat operation. And this is some real work. I'm not going to talk so much about it either. But the important thing here, this is using a different model in which the free energy surface now is defined as a basically a Landau polynomial like this. You feed the calorimetry data to kind of model, and then you can, from the width of this transition relative to the height, you can estimate the barrier in thermodynamics. This is not a kinetic barrier, by the way. This just has to do with the probability of the states at the top. Okay, so using that analysis also, you could look at many different proteins, which we did for 15 proteins that they have very high quality data from calorimetry, and they also have kinetically determined rates. And so we did the fits. This is for core shock protein, for example. The model worked well for all of them. And when you're plotting now the barrier, thermodynamic barrier you estimate from this analysis compared to the rate that people have measured experimentally, you see that there is a pretty good correlation, especially in this regime over here, which is when the barriers are very small. Okay. So what you get from here is basically the same kind of behavior. So these are two different ways of looking at experimental data. In both of them, they tell you that barriers for folding are relatively small. I know, typically in the order of a few KT at most. Okay. And again, this, which from the point of view of simulations is relatively easy to figure out, from an experimental point of view was not clear. And people said, because I can fit data to a two-state model, that means that my protein is a two-state system. And the problem is a two-state model is very permissive. So you can actually fit almost anything. You can even fit 300 different behaviors together to a two-state model, provided that the model is flexible enough. Yes. That's right. Anyway, so this is just to say that, later on we show that you can actually make this kind of analysis better by combining multiple models and then do a Bayesian analysis. Oh. Thank you. All right. So basically, this way, using multiple models, you can eliminate whether you like one particular model or another one. All of them are free energy surface models, so they're based on projecting the surface and doing the analysis. But they produce different surfaces. But at the end of the day, the barrier heights they produce are consistent. And using a Bayesian analysis, you can actually get a model-independent estimate of the barrier from calorimetry experiments. Anyway, so what all of that tells us is, okay, it really does make sense to try to look at protein folding using standard equilibrium experiments with a technique that has atomic resolution, because you're supposed to be able to get a lot of information out of this. And in principle, try to figure out a way of mapping this folding interaction networks, which is what I'm going to be telling you about. So what I will discuss now is some particular experiments on one protein. This protein is called GPW. And we like this protein because a lot of the criticism regarding the experimental downhill folding work is that, okay, you're working with a protein that is, 40 residues, it's teeny tiny, it's not even a protein. Who cares? It's also a little domain. Maybe you cut the domain and you destroy the cooperativity of the system, so what you're seeing is an artifact. This protein is nice because it's a single gene product, so it's not a domain. The protein is like this in nature, okay? It's also 68 residues, has 2 long helices, and it has beta structure as well. It's not only helical. And this is, larger than Ci2, larger than HH3. So, there are not any of those issues don't appear anymore, or shouldn't appear, all right? So this protein, as I say, we studied it in the lab. We found using laser T-jump methods, which I didn't have time to go over yesterday, but allow you to look at processes that are pretty fast. We saw that it's a fast folder. And this is interesting because based on the contact order prediction, this protein should be relatively slow, but it's fast. At the folding temperature, which is around here, is on the order of 8 microseconds, so it's really fast. Okay, so we can use the same type of model to analyze the kinetics. I'm not gonna tell you how you do that, but all these fits are actually from the analysis of with this model. Here you have the relaxation rate as a function of the temperature, well, inverse of the temperature because it's an Arrhenius plot. And then when you do the analysis, you see that even at the midpoint, the protein, it has a barrier, but it's a very small barrier. It's on the order of 1 kT. So it seems like an ideal system for this. We also did the equilibrium analysis by calorimetry, and the green is what you would expect if the protein were perfect to state-like. What you get is something broader. And this doesn't seem like much, but from the point of view of calorimetry, this is a big effect, let me tell you. Okay? So it tells you clearly that the protein is much less cooperative than you would expect for two-state. We also look at the equilibrium using some low-resolution techniques like fluorescence and FTIR and circular dichroism. And we could see already signs that the behavior is a little bit different depending on what you're looking at, right? So probes that look at the backbone seem to have a higher folding temperature than probes that look only at the tertiary contacts of this particular residue, this tyrosine. Okay, and this is the analysis that I was describing before for the calorimetry, and this analysis gives you something like this, in which you see that at the midpoint there is a barrier, but it's teeny tiny. And this barrier that we get is smaller than in the kinetics. This is something commonly found, and this is actually something that theoreticians and people doing calculations also find, right? Because what we have here is not a kinetic barrier. It's just the distribution of probabilities at this region. And of course, the connectivity in kinetics is not perfect, probably because this is not it's a good order parameter, but it's not a reaction coordinate. Okay? But still, the difference is relatively small because we do get a teeny tiny barrier. It's of the order of 0.3 kT versus 1 kT. So no huge differences. So from the point of view of energy landscape, you would say this protein is nicely smooth in terms of the landscape and so on. Not much frustrated, as Diego likes to look at. Okay, so this protein is a good model for doing these experiments. Now I'm going to go and talk a little bit about NMR, very basic, very quickly. As I say, if I'm getting really boring, let me know and I'll go a bit faster. Okay? But there are a couple of points that I like to make that are important to understand the limitations of the experiment, in a way. Okay, so we're going to look at this using NMR spectroscopy. And of course, the first thing to know about NMR is that, in order to do NMR, you have to have atoms that have a spin. And that means they have to have an odd number of neutrons and or protons, okay? So in protons, we have protons, which obviously have spin. And we can look at NMR in any protein by looking at proton NMR. But also, if you can isotopically label the protein, you can also, instead of having carbon-12, you can have carbon-13, which also has spin, and nitrogen-15 instead of 14, which also has spin. There are procedures to actually prepare proteins with this, and this is the way you actually do high-resolution NMR, and you get structures and all these things, and you look at dynamics. Okay, you can also do this with fluorine, but not the useful for proteins. Anyway, so all the experiments that I described before on BBL were done only using proton NMR, because we were making the protein chemically. Everything that I'm going to be telling you now combine all these things, because we are using a protein produced by recombinant means, and we can isotopically label it. And then these probes are more accurate indicators of structure than this one is. This one is too sensitive, and it depends on many other things. Anyway, so when you do NMR, what you do is basically you apply a magnetic field, which is typically in the order of 10 to 20 Tesla. And what that means is obviously all your spins are precessing at some frequency, which is determined by this magnetic field times this thing called the Gyromagnetic Ratio, which is basically specific of each nucleus. Right? So protons have one, carbon-13 has another one, nitrogen-15 has another one. And this is what determines when people say, okay, I have a magnet that is 600 megahertz. What they really mean is they have a magnet that is 14 Tesla, which in that magnet actually protons precess at 600 megahertz. Okay, so that you get an idea, because sometimes that's confusing. Anyway, so then obviously what happens with NMR is that, because you are looking at spins, it's basically very small difference in energy, right? Because we are in the radio frequency range. Nothing to do like with typical absorption spectroscopy, things like this. So what that means is that you need these very high fields in order to produce a very small difference in population between the ground and the excited state. which is this. Obviously, this is proportional to the field, as I said. And still, even with these huge fields that you use, the sensitivity is very low. Because here we have population differences that may be 0.1% or things like this. So basically, most of your molecules are not doing anything for you. And that's why you have to use typically millimolar concentrations to do these experiments. And that's important because it's a high resolution technique, but it's extremely low resolution sensitivity technique. And that's important, okay? Because one of the limitations of this approach is these concentrations. Not every protein can do that, can get into there and behave well. Now, if you think about this, obviously the proper way to describe NMR would be using quantum mechanics, but you can do a semi-classical description in which basically what you say is now you apply a field and your spins are going to either align or in the opposite orientation. Obviously that makes the difference in energy. And also that means that you have basically a net magnetization on the axis of the field that you have. And all the spins, as they will be precessing, rotating here, depending on this gyromagnetic ratio. Okay, so the way you do the experiment now is, obviously you have that, you don't see anything because you have an equilibrium system. What you do is you apply a radio frequency pulse, okay? And you do it in orthogonally to the magnetic field. So, in this plane, this is the z-axis, so this is the xy axis, okay? When you apply a pulse in one direction over here, what happens is that you flip some of the spins, and once that you flip them, obviously they are going to be rotating, and they are going to be relaxing back to equilibrium. And in this process, what happens is if you have a probe over here, that different probes in different positions, you can record this precession frequency as a radio frequency signal in which the spins are moving until they relax, and then the signal disappears. Okay, so depending on the intensity of this pulse, you can produce something that people call a 90-degree pulse, which is when you put 25% of the inverted population into this plane, and that's when you see the maximum signal, which would be this situation, right? So you flip the magnetism over here, and then you have a maximum signal. If the pulse is a bit longer, you go over. You can do a 180-degree pulse, which is you completely invert the population to the other side, and you don't see anything, or you can even go backwards, okay? Because remember, the populations go in the z-axis, and what you're recording is in the xy. So that's the characteristic. So anyway, this kind of fluctuating field looks like this. It's called an FID, which is a free induction decay. And of course, what happens is you see these oscillations, which have to do with the frequency of the system, the precision of the protons or the carbons, whatever it is, and it decays because the system eventually reaches equilibrium, so you lose the signal, okay? So now you do Fourier transform of this, and this thing, which is in the frequency domain, goes into something like this. This is in the time domain, and then you get the frequency domain in which you have the intensity, the frequency, typical frequency of this, okay? Which is the values that you're interested in. This is Lorentzian, by the way. Anyway, so so that's the idea. So the way you do the experiments now is typically you have to wait for some time until the system is in equilibrium. You apply one of these 90-degree pulses. You want 90 degrees because it's when you get the highest signal. Then you see this free induction decay. Now you do Fourier transform, and you get a spectrum like this. And the spectrum is shown typically in these things called ppm's, which is a little bit weird, right? So what is this ppm's? Well, it's very basic definition. And the fact of the matter is that even though all atoms of one kind, let's say protons, precess at 600 megahertz, there are small differences between them. And these differences depend on the magnetic field they have around. And this is what tells you something about the structure, okay? So these differences in magnetic field come from the fact that you have other spins nearby. They are very small, and this is why you define them in parts per million. So the way you actually do this is define this chemical shift, which is basically the frequency of the particular proton or other atom that you're looking at relative to a reference, right? So it's the reference minus your measurement divided by the reference multiplied by 1,000,000. So that's what these chemical shifts are, okay? And they are useful because, as I said, depending on the environment, they will have slightly different frequencies, and this will tell you a lot. They tell you a lot about the chemical properties of the proton, because depending on what other atoms are nearby by chemical bonds, so that means that it allows you to assign the spectrum to see which frequency corresponds to what atom, and also depends on the structure. And this is why it's a useful structural technique, because depending on how this changes from one proton to another proton, you know more or less how the structure is changing. So this is just a list of some typical chemical shifts for different types of protons, depending on the environment. For example, if you see, methyls are typically over here, very close to the reference. When, the more polar is the environment, the chemical shift shifts more upfield, which is this way. And what that means is this allows you to have a fingerprint for every type of atom and do the assignment, but also it gives you these structural probes that I was telling you about. Now, let me just tell you something more about the NMR. I will probably not spend too much more time on this, but this is also important, which is the relaxation process. Okay, NMR is very different to other types of spectroscopy because you are in this radio frequency range. What that means is that normally in NMR you don't relax by a spontaneous or stimulated emission, which is what you find in other spectroscopies. Here, what you have is relaxation that is affected by basically it's called magnetic spin relaxation, is affected by the environment. And typically, you can define two types of this relaxation. You may have seen people talking about this. One is called T1, and that's basically the longitudinal relaxation. It's basically on the z-axis, and this is really what it brings the system back to equilibrium, right? So that means that when your system is, you're pushing it down, it takes some time to actually relax back, okay? And this is what determines how fast, how quickly you can repeat the NMR experiment. And then there is this other one called T2, which is kind of more obscure, but it's more interesting from what I'm going to be telling you about, which is called the transverse relaxation, or T2. T2 for the time, R2 for the rate. So this is basically a relaxation rate, or a relaxation time. This one basically has to do with losing coherence on the XY plane, okay? So you can see it over here. So what happens is once that you tumble your spins and you start seeing the process in the XY axis, what happens is you start with everything basically synchronized because you put all the spins at the same time, but each of the spins is going to be rotating with a slightly different frequency because they have different chemical shifts. And what that means is that they're progressively losing coherence. And when they lose coherence completely, you don't have a signal anymore because you're looking at this on the XY plane. So the system is not at equilibrium yet, but you don't have a signal anymore. So what that means is that your free induction decay is not telling you about the equilibration to equilibrium. It's actually telling you about the loss of signal in the XY plane, in the transverse plane. And this has implications, first of all, because it depends on the molecular tumbling. I mean, you can imagine, not only because the chemical shifts are slightly different from one another, but also because the molecule may be tumbling as we go, if the time scale is equivalent. So, that means that the coherence is lost as well. And there is one more effect that this has, which is interesting from the point of view of doing the experiments, which is this type of chemical shift anisotropy or effect that is produced by that also is affected by the possibility of having exchanging conformations. Okay? Now you imagine that in the time scale of doing the experiment, making the measurement, if there is something going on so that the protein is changing from one state to another one, and these two states have different chemical shifts, you're going to induce another loss of coherence, right? Because now your chemical shift is changing, so your rotation frequency is changing as well. Now, if you think about the time scales here, typically chemical shifts are on the order of 1 part per million. So you have a 600 megahertz instrument. That means that you're roughly, thinking about something that is half a hertz. Okay. And what that means basically is that any process that has rates in the order of the milliseconds or so is going to be affected in this process. And what that means is basically you can define two different regimes. One is the slowest change, in which whatever happens is so slow that it doesn't affect the measurement, so it's much slower than 1 hertz. The fastest change, which is when anything that is much faster than 1 hertz, it's going to be happening so that you have a time average signal. And then you have this other regime in which basically you can get kinetic information from the broadening of the signal. All right? Okay, so this is important, as I say, because in the experiments that we do, we are going to be changing the folding properties of the protein, and that means that there's going to be a change in populations. And if this change in populations is in this regime, we wouldn't be able to see anything because it will be too broad. If we do it in this regime, we will be seeing two different populations, one corresponding for the unfolded state, another one for the folded state. And if we do it in this regime, and it will be very hard to do, but it's possible, and we've done it. But this typically will be for proteins that fall very slowly, and what that means is that they will have higher barriers, so there will be less to see. And this is the regime that is more interesting for us, because in this case, you have a fast process, you get a time average signal, and this is actually coming from the populated, the ensemble of conformations. It's a weighted average, all right? Okay, so that's as much as I'm going to be telling you about, so let me just skip this. Now let's go to the experiments. Okay, all right, so this is what I was telling you about, so about the difference in frequency. So this depends on the difference in ppm's from one to the other. As I said, the typical area in the order of 1 ppm, roughly. And we want to be in this regime, typically, okay? And this is why, fast-folding proteins are useful for this. So that's what I have here. So their folding rates would be faster than 100 microseconds. So you remember the one I was telling you about at the midpoint was 8 microseconds, so it's well here. You obviously need also a system that is fully reversible in thermal unfolding at millimolar concentrations. This happens to be a very demanding criterion because many proteins may be reversibly folding and unfolding by chemical denaturation, but when you do it thermally, they are much more prone to aggregate, okay? And the idea of the experiment, as I say, is a very basic experiment. You are going to be assigning the protein, so you will know each chemical shift, to what atom it belongs to. And then you will be doing experiments at different temperatures, and you will be recording the NMR spectra, and then you will get the NMR, the chemical shifts, and you will compare them for the different atoms as you go along. And the way to compare them is to use a reference, which is what people call the random coil values, which are the chemical shifts that those typical atoms have when they are actually in a single amino acid with no context, okay? So you measure all of them at the same time because they have different frequencies, so they look different. So let me just, so this, for example, is what people call an HSQC experiment. It's a correlation between the nitrogen 15 of the backbone and the proton of the same, okay? So each of these cross peaks corresponds to one particular nitrogen proton amide for one amino acid. Okay, so you see they are all different, right? So you do the experiment, you can see all of them. There is a procedure to actually assign them, and I have the slide, so you're interested later on, I can tell you. It's actually pretty smart, so the way you do this, you do several experiments in which you couple different types of atoms depending on their chemical properties. So things that are close in distance, things that are close through chemical bonds, and by doing all these coupling experiments, you can track which signal is which. And at the end, you come up with an assignment, which is what I have here. This assignment is, for every particular cross peak, I know which amino acid belongs to. So this is, for example, alanine 37 on the protein. This is alanine 24. This is threonine 36. So each particular peptide bond now has a probe here. I can do the same thing for the C beta, for the C alpha. I can do that for the alpha proton. So for each of those chemical shifts, I can track. This is just showing one example of those, okay? So this is the spectrum in one condition. I believe this is at 20 degrees. So once that you have the assignment, the experiment is relatively easy. I mean, the assignment is hard to do. You have to do multiple three-dimensional experiments and things like that. But once you have the assignment, the only thing you have to do is repeat the experiment at different temperatures, which is what I'm showing over here, right? And then you track the signals. And sometimes the tracking is very simple because it's not moving very much, as you can see over here, for example. But then the things move more, here they could be somewhere over here, and in that case you will have to do the three-dimensional experiments again to reassign and make sure that you are not crossing things, okay? But in principle, as I say, it's difficult to do because it's involved, but the procedure is straightforward, all right? So this is what I'm trying to represent here. This is the same spectrum, and now you do slices. I mean, each of these slices is the same spectrum at different temperatures. So you can see, for example, the signal X is not moving much, and then it starts to move, and then it starts to get closer to these other ones. As long as nothing is crossing, it's easy to track. But in the moment that something gets to cross, then you have to do the 3D experiments again. So normally the way you do the experiment overall is you do the isotopically labeling first, then you do the 1, 2, and 3D NMR experiments to do the assignment. All of these are all these funny acronyms that tell you all these couplings that I was telling you about. All right. And as I say, we could go through that, but this is not an NMR structure determination course anyway. And then, as I say, once that you do that, then you do the same experiments, or at least a subset of them, at different temperatures. And this is really where the information about folding is, okay? Because what you're doing now is looking at the chemical shifts, the environment, the electronic environment of every atom on the protein as you're unfolding the protein. Okay? So when you do the experiments, you get things like that, which basically means it's a mess. So typically when you do the experiments I was showing before, they look like nice sigmoidal curves, which is what you expect for a first-order light transition. Obviously here we have probes that are so sensitive that any slight change in the environment produces a perturbation. These perturbations are interesting structurally, but they are not easily interpretable, okay? So you have things that look like this. This looks like a nice first-order transition, right? But then you have things that look like two transitions or even three. Sometimes you get something funny like this. You don't really know what to do with that from the standard way of thinking about this, right? So in order to take and this is probably why some people try these experiments even though they didn't seem to be, and then they give up because the people wouldn't know what to do with this, right? You can, how do you interpret this? So the idea here we had is that in principle, the first thing that you need to do is to classify them and then try to do some way of sorting them and then try to cross-correlate them. That's kind of, in general, the approach, okay? So the first thing, so we classified these ones in basically things that look like single transitions, things that look like double transitions, which is like this or like that, and then things like this or like that, we put them in order, okay? So that's the first thing we did. Now, the next thing you have to do is, okay, and this is a typical criticism when you do these experiments. People, because they, when you have a two-state minded view, you would say, I look at all of these things, and what I'm seeing like this is not telling me anything about folding. It's some kind of local fluctuation I don't care about, right? And then what I will do is I will prune this data until I have something that looks like all of them very similar. People will actually have they will justify themselves saying, that's the global transition, and everything else is kind of local fluctuations, all right? But the first thing that you need to show to people, I think, is that all this data is actually important, and all of this contributes to the ensemble average, which seems obvious from a physical point of view, but as I said, it wasn't. So the way you do this is, you take all of this data together, all of it, including all of this, and you just calculate a first moment of it. Obviously, you have to do it in an intelligent way because they have different signals. Some of the chemical shifts go down, some of them go up, depending. But if you do this, using singular value decomposition, for example, or some technique like that, something simple, then you get an average curve for all the species that we have there, and we have like 200 and something curves. The average is this blue curve that looks like an isigmoidal, right? And not only is an isigmoidal, it's also very, very similar to the curve that you get with a low-resolution technique, like CD, right? So this is a very important control because it's telling you that all this complexity is really associated to folding. It's just you're looking at local perturbations, but they all contribute to the process, okay? And you can do the same, and you can even go further and do a little test. You say, okay, rather than putting everything here, let me just do this in two sets. I will look at the curves that look only like first-order transitions, and then you get the yellow one. Or you can look at all the ones that are two-state-like, double transitions, and then you get this other curve. So none of them are as similar to the low-resolution technique as the global thing. So basically, no surprise, right? So you look at the global process with a low-resolution technique, you see something that is similar to the average of a high-resolution technique. This seems like trivial, but as I said, this is a very important control from an experimental point of view because it does tell you that what you're looking at is actually folding, not just some other funny thing that you should forget about. Okay, so now that you've done that, then basically we classify all these curves, and these are some examples. So we did a cluster analysis to put them in groups that were more similar to one another. And the clusters, these are representative examples of the clusters. This is the average behavior as it will look like for that particular probe, so with the baselines and all of this kind of thing. So you can see that they are all more different or more similar. So for example, this one here is very good representation of the average behavior. So all the probes that are in this cluster are giving you something that is similar to the global folding process. all the other ones are giving you something more particular. You can plot all these clusters into the, kind of a topology diagram of the protein. And the first thing that you can see, and this is something that people also find surprising, is that there doesn't seem to be like a nice separation of colors here, right? So like, for example, a helix unfolds first, and then it comes to the second helix. We don't see much of that kind of element by element picture. We see something that is much more fine in detail. For example, I mean, you can see a lot of the, the average cluster over here in this helix, but then nearby you have other atoms that are seeing something slightly different. Okay, so the next thing that you need to do here is to do some other analysis. And here is when we go to do a cross-correlation analysis. And this is a very simple analysis, really, which you have here. The idea is that if you have curves like this, the implicit idea is that if we see some behavior that is very similar, that means that they are thermodynamically coupled, right? If we see things that are very different, that means that they are thermodynamically uncoupled. So they actually are seeing different type of processes. So by doing something like this, you can define some metric on how similar these curves are, and this has to do with basically how the probability changes as a function of temperature. And then you can, once that you have this metric, then you can define something like this, in which this is like an RMSD calculation, right? And what we have here is some kind of normalization. This is again important in experiments. And this normalization takes into account the probability of things being similar by random coincidence, right? That's what this term is. And this one is also the probability of seeing something similar because you have noise in the experiment. So this takes into account experimental noise. It takes into account random occurrences. This is your experiment. So it's basically a ratio. These are, a normalized RMSD. You put this in a log scale that gives you this index that is telling you how cooperative the process is, right? So if this number is high, that means that, all the curves are very, very similar. Your system looks like two-state. So it's similar, it's close to an all-or-none process. If this is 0, what that means is that the process is completely random. There is no coupling. So you're looking at things that have nothing to do with one another. And if you have any number in between, that means that you have a process that is cooperative, but with a limited cooperativity, and you can determine how much it is, right? And that's, for example, for BBL, which was this global downhill, we got a 1.3, so very, very, very small cooperativity. This is in kind of RT units in a way, roughly. For GPW, we're getting something that is about twice, but it's still relatively small. Okay, so that is for the average. Now, the other thing that you can do is you can look at this process on a residue basis. And we want to do this on a residue basis because at the atom level, probably we are seeing too many local things. But if we have multiple probes, atomic probes for every residue, we can actually look at all of them together and see how they compare for different residues. And that way, we can get something similar to a contact map, but for couplings and folding as a function of temperature, okay? And this is what this kind of thing looks like. So this is the same type of calculation I was showing before, but now you do it on a residue basis. So you take all the probes for one residue and you compare them with all the probes of another residue, and you see how similar they are. So it's the average correlation between all the probes for a residue pair, okay? So, obviously the diagonal will be the single residue. Everything of the diagonal is the couplings. In the way we plot this, blue means completely uncoupled, and the dark red means strongly coupled. And it's a relative scale anyway. But what this tells you is, okay, you can see what what residues seem to have a more correlated or more coupled unfolding behavior, okay? And this is obviously something that you can compare. This is the matrix for GPW. So you can see there are some couplings that are very strong. Some of them are local here. This is actually helix 2. This is helix 1. This is the hairpin. And you can see that there are some couplings that are longer range. Now, what you can do in order to try to make sense out of this is overlay it with the contact map. And you do this by putting this in green and this in red. And this looks like that. So what you get from this comparison is you get red dots, which means these are atomic contacts that don't seem to have much coupling in folding. So then you get green dots, yellow dots, which are contacts that seem to have a lot of coupling in folding. And then you get green dots, which are the most interesting ones because these are residues that are coupled thermodynamically in folding, but they are not in direct contact. But you can always track these green dots to maybe secondary or tertiary couplings, right? Things like, you have 3 this one is not in contact with that one, but they are in contact through something in between. So you can actually do that kind of math, and it kind of works. Okay, so as I said, this is the data for this protein. Now, obviously, this is interesting because it gives you a lot of information about, the folding interaction network, so what things are more important. So, for example, contacts that are red here probably would imply that, these are contacts that happen, they're adventitious, right? They happen because the structure is formed. Once you form the structure, there are, things that are necessarily in contact. It doesn't mean they have a strong interaction. This is important from the point of view of doing kind of Go models and things like this. So this is probably why, you can go as far as you can go with Go models, which is pretty far, but not as far as some of us would like to go, right? You can look at the same data and plot it from, do some kind of network graphing. And what that shows here, and this is, this kind of fish structure is basically telling you what are the most important residues in terms of the coupling of the structure. And you can see them all here. This is the hub, which has all the important connections. And they happen to be in these two regions, right? It's the connection of the two helices here in this end, and these two loops, the stuff that seems to be most important. So, in a way, it makes sense, because you look at this protein structure, it's fully antiparallel. So you have one helix, loop, hairpin, turn, strand, turn, strand, loop, helix. So in the moment that you take the two loops connecting the helices and the strands, you close them and you put them together, that's it, you're folded. And that's pretty much what this is showing. So it does make some structural sense, right? Okay, so as I said, the nice thing from an experimental point of view is you can get information that we couldn't get any other way. The other nice thing about this is that it's something that can be compared with simulations. Okay, and that's what we did. So we team up for this work with the show people. So, Angel was showing Anton yesterday, I believe, right? So that's the same picture. This is all stuff already. So it's the old Anton or the new ones. And this is the simulation they did on GPW before we were, this is not working. Ah, yeah. Okay. And this is coming directly from the show, so let me just, it's interesting. I cannot control the volume. Anyway, you're going to have to listen to the show talking about this. Anyway, so this is the simulation they did in which they were seeing folding and unfolding of GPW. They did this without knowing our data. Actually, they did it before we finished our experiments. And then when we learned about this, we got in contact and said, okay, why don't we look at these two things together and see whether they make sense, right? So they were seeing folding and unfolding. They ran a 230 microsecond simulation at the time. So anyway, so I'm going to skip that. So basically, this is the analysis they got from their simulations. So they did two simulations. So they did the standard simulation of the folding temperatures in folding and unfolding events, which is what this is. And they also did a parallel tempering simulation, which is similar to replicate change, so that you could compare with the thermal denaturation experiments that we did. All right. So in their analysis, that's what this is. So they saw unfolding, they saw different clusters, they classified them according to their criteria. And they did find some particular clusters in which there was a symmetry issue, right? Which are this, they're called M because they are mirror images. But in general, all of this is probably what is more relevant for the process. And first indication you get from the simulations is not a two-state system, it has complexity. Some of the complexity correlates with what we see in a qualitative way, right? Because you see, for example, I didn't point this out, but the helices seem to be highly correlated with one another, right? Each one with each other. And that's what it appears over here. You can see the helices in these clusters are still kind of formed. Okay, so this is interesting from the simulation. If I was a simulator, I would spend a lot of time talking about this, but since I'm more like an experimentalist, the important thing for me was to try to compare the two and see how they look like. So that's what this slide is. The first thing is obviously the simulation picked the native structure really well, or relatively well. And this is an important issue. I didn't point this out, but this protein had been solved, the structure had been solved by NMR by another group many, many years ago, well, many, in the late 90s. And the structure is different from this. And actually, these show people had simulated this, and they couldn't get the native structure, so they were kind of worried about it. And they told this to us in a meeting and said, we don't, and we were actually having a similar issue because we had solved the structure of this protein and we found that the original structure was wrong. And we were about to publish this, and then we told them, actually, the original structure is wrong because they misassigned some critical NOEs in this defining the position of the hairpin relative to the two helices. And our new structure actually is very similar to the simulation, which is what this is. And it's quite different from the original structure. So that was kind of interesting. That's another control, like similar to what Angel was saying yesterday. Now, you can compare the kinetics, and the way the DSO people were estimating the kinetics was by doing an autocorrelation function analysis. And in this case, this is the plot for the autocorrelation function for the helical residues. Okay, so the helical hydrogen bonds, really, to be more accurate. So they were getting a correlation, time of 4.6 microseconds, and experimentally, you remember I showed you this before, is on the order of 8 microseconds. So it's pretty close, a factor of 2. They worry about this. To me, a factor of 2 is really remarkable because also here, in difference to what Vijay Pandey was doing originally, he is not defining an arbitrary threshold in RMSD to decide when you're folded. This is just an autocorrelation analysis, so there is no tweaking here. Because in the moment that you define a threshold in RMSD, changing the threshold, you can change your rate by a factor of 100,000 very easily, right? By just changing this a little bit. So those initial calculations were a little bit of problematic that way. This one doesn't have that problem. Now, so this is for the kinetics. Now, if we compare the equilibrium, so what we did here is we took all the simulations from the DSO group, and we used programs to calculate chemical shifts from the structures. They're not perfect, but they work kind of reasonably well. So we could simulate chemical unfolding curves, chemical shift unfolding curves from the simulations directly. And we could do exactly the same analysis that we have done with experiments. And this is the nice thing. So we are not interpreting signals. Well, we are, in a way. But we are using an external interpretation, not our own. Okay, which is these programs like CS Rosetta, which basically, you put a structure and it gives you chemical shifts. You put the chemical shifts, it gives you a structure. So that kind of thing. All right, so in this case, we're plotting here the average chemical shift as a function of temperature. So in blue is the fit to curve that I was showing in blue before. And in red is what you get from the simulations. And it looks outstandingly in agreement, but obviously we're cheating here because the temperature scale is different. Okay, and this is the most important thing. There is a slight difference in folding temperature as well, but that's very small. So, but I think, so they got the folding temperature within 5 degrees. Now the problem here is the temperature scale is compressed, or it's actually expanded in the simulations by 3.15. So you can see here it goes from 280 to 440, here it's from 310 to 360. So when you compare them well, it's when you compress this. And what that means is that the temperature dependence in these force fields is not good, and we know this. It may have to do with some of the things that Angel was saying yesterday. It may have to do with polarization of the solvent. Well, I don't really know. Maybe some people here have ideas about this. CS Rosetta, Talos, and Camshift. We did the 3, and we looked at all of them. They give you something very similar. Okay. So what this technique tells you is that, okay, this is a very good experimental method to actually test force fields for this particular problem, right? Which is there and has to be fixed. Now, if you look at, the next thing that you can do is you can do the same type of analysis, but rather than looking at the whole protein, I'm going to be done in a minute, you look at this by helices, right? So by secondary structural elements. So you look at all the chemical shifts from one helix or from one strand, so on. And when you look at this experimentally, you see there is differences, but the differences are not that much, okay? Because everything averages out in a way. When you look at this from the simulation point of view, you see similar pattern. Actually, the trends are similar, but the spread is much larger. And the spread is larger with the same when the scale is compressed. So this is a relative effect, okay? It's not the same. And then the next thing you can do now is you can look at these matrices. This is the experimental one I was telling you about before. The circles here represent the contacts, and the colors represent the couplings. And this is the simulation. And what you can see is that overall, okay, you say, it seems to be quite similar, in a way, remarkably similar, when you look at this at a glance. But if you start looking at this in detail, you see significant differences, right? For example, you see right? So what that means is that there is more complexity in the experiment than in the simulation. The simulation seems like the helix goes all together in a way that is more cooperative than in the experiment. And the opposite is true for the long-range contacts. So we see much more couplings in the long range than in the simulation. So the simulations seem to have a system in which the local interactions are too strong and the long-range interactions are weaker than in the experiment. And that means that the cooperativity here is less than here at the protein level, but is more at the secondary structure level. Okay? And then at the end, you also start to see, okay, so there are things that are common. For example, you can see that the hairpin is very uncoupled in both. And this is something that we also seen by relaxation dispersion experiments by NMR, together with Luis K. We published another paper on that. And also the long-range contacts here are also highly uncoupled on both. But if you look at the pattern of important contacts, it's quite different, right? The contacts that we see experimentally to be very important are not the same as they see in the simulation. And you can see it over here. So these ones versus those ones. And, and you can see that comparison here a bit more clearly. So the trends in general are similar, but besides the overall effects, there is a lot of detailed information here in this data that could be used to try to refine the models. And I guess we are now doing, with this I'm finished. We are doing experiments like this on other proteins. We have like 3 other proteins that we haven't published yet, but we have the data. And what I think it would be nice is if we convince people to use this data, I mean people like you guys, to try to use this for refining models, whether it's coarse grain models in which you can actually make up the force field the way you want to try to reproduce experiments accurately, or get atomistic simulations and try to get into charm or amber or whatever and try to see what you need in order to get this to be better. Not only at this level, but even at the coarse level that I was talking about before. Okay, this and that. Here? Yeah, the letters tell you about the average color of each of these going through the whole thing. Red means very strong, blue means no coupling. Or coupling as close to random. Okay. At the amino acid level, I mean, you can see here, there are some similarities. For example, you see high coupling there, right? Which is this region, right? And you can see very little coupling over there, which is this region here. Also, to some degree, with some, I mean, there is like more detail in the experiment and in the simulation, but here you can see these ratios are coupled and they are coupled there as well. So there are some trends, definitely. They had to do with the topology of the structure, and they probably had to do with the sequence more than the chemical properties of the atom. Because remember, what we're doing here is we are getting multiple probes. And so we are looking at the C alpha, the C beta, the nitrogen, amide. So all of those, in a way, average out all the details, and they give you something about the global picture for the residue. And I actually think that's an important feature of this, because if you look at the atomic level, then you're really looking very, fluctuations that are too uncorrelated. So, you get too much noise. This way, it's probably a nice balance. At the residual level, I think it works out reasonably well. Okay, so I'm done. Sorry, I went a little bit over time.